Yes, great. Yeah, so this talk in a way nicely connects to what Peter presented. So I'm Alex Schoenhuth. I'm from Amsterdam. I'm from the Centrum Wiskunde en Informatica, which is Center for Mathematics and Computer Science. And I will talk about how to discover somatic variants in single cells, like from a different. I will talk about this from a different angle, and like really focus on the mathematics and computer science. I hope that some of you are interested in this, given that some of your supervisors are interested in it, for what I understand. Anyway, so I will also tell a little bit of a story. Peter talked about the recent challenges in single cell sequencing here. What he did not talk about so much is kind of the whole story about like what the challenges are in next generation sequencing, variant calling in general. So you can consider this here a little bit of a history lesson as well. I thought, like, with all the students in this room and this being part of an international training network conference, I could do this. So I will first talk about how to discover somatic insertions and deletions in bulk, in bulk. And if you understand this part of the story, you're much better able to understand the second part of the story, which is connects to what Peter talked about. Yeah, let's go into this. How to discover somatic indels from bulk sequencing, and we start with this picture here, which I think indeed many of you understand immediately. What you do in bulk sequencing is you analyze a control genome, which is a healthy genome, and a cancer genome that emerged from the control genome by the application, quote, of somatic deletions to the control genome, and then turned into a cancer genome. And you have to distinguish between germline variants that apply for both control and cancer in comparison to the reference genome, and for somatic deletions and insertions that only apply for the cancer genome, and it drove that genome into this kind of aberrant kind of thing. Yeah. Here, so next generation resequencing. This is the general plot. You extract donor genome DNA. If you do bulk sequencing, this is from like 10 to the 3 to 10 to the 6 many cells at once, and if you do single cell sequencing, this is only from one single cell. Then you break it into fragments, and according to still popular protocols, you generate paired end reads. You sequence the ends of that, and then you align those reads against the reference genome. And this is also what you do also here in single cell sequencing. It's also part of the game that Peter talked about. Yeah? And then you try to spot the single nucleotide variants, for example here, by comparing the aligned data with the reference genome. For example, you have a T here in the reference, whereas in the genome you're investigating, you have a G. So the reference genome isn't but a coordinate system. Anyway, it's not so important. And the data we're dealing with are paired and alignments. Yeah, this picture here we're dealing with in basically all of what follows. Yeah, and it was kind of hiddenly also used in Peter's work, of course. The first point is this is largely also uncertain data. So we had this very lovely picture here. Yeah, like you had a coverage of 6 and all aligned very well. But here, this is reality. This is the IGV integrative genome viewer, and this picture reflects here is the reference genome, and this is the paired end alignments you're watching. And this part here of the reference is duplicated, so it appears another time somewhere else in the genome. And then all the reads that you broke out there, all the fragments you broke out there, also align here, and vice versa. And you do not really know where they're from. Yeah? So the placement of those fragments, these paired end alignments, are uncertain data, as you call it. Yeah. And so my passion, in a way, is to deal with this and to have a real statistical count for these uncertainties, and to really drive the performance rates of the callers and so on to the extreme, I hope. And this is the fun I've been busy with in the last couple of years. Okay, first we would like to call deletions and insertions, and they are the uncertainties become particularly difficult to deal with. So you have a reference genome and the sequenced genome, I mean the genome you're investigating. This part, which is of length 12, is missing. And then you break out the fragments such that, after sequencing, the deletion affects the internal part. So if you then align it, you see, okay, the alignment is longer than a fragment. The problem, though, is you can measure the alignment length by doing right reference coordinate minus left reference coordinate. However, you do not know how long the fragment was because you you forgot to you did not sequence the internal part. So also you do not know how long the fragment really was. So you do not know whether this is too long or whatever. So all you can do is to compile a fragment length distribution, which you can do from the Paradigm read alignments, and any kind of standard good state-of-the-art read aligner will give you this. Yeah, and it has a mean and a variance, and then you can imagine, okay, imagine that you had this deletion of length 12, and then you consider how are the alignment lengths distributed under this assumption. And you end up with these two distributions. This is the original fragment length distribution, and this is the alignment length distribution, given that you assume that there was a deletion of length 12. And then you can put the alignment length here on the x-axis, and you can compute the probability that it really stemmed from this distribution versus the other one. And then you have some, straightforward statistical considerations. And then in the end, you can, with certain kind of levels of certainty, you can conclude there was something or not. So actually, it's only really still today, it's only really certain if the deletion is kind of at least 150 base pairs, because then the blue distribution is kind of far out to the right, and then when you have an alignment length of this length, then it really doesn't really touch the red distribution anymore, and so you can call it. Yeah? All right, there's other evidence you can make use of, and this is the split read evidence where the deletions or insertions or whatever affect the sequence parts of the reads. However, I mean, this looks like much more certain data. However, I mean, aligning sequences is still like the most traditional challenge in bioinformatics, and still not fully solved. And in the next generation read aligners didn't do a particularly good job in this. Actually, they introduced no improvements whatsoever in a way in this respect. So you are dealing with possible well-known effects like gap wander, gap annihilation, tons of publications, all dating back to the, I don't know, early 2000s, whatever. It's still a challenge. Yeah. Nevertheless, I mean, you have the evidence and you can deal with this, and it works actually fairly well for deletions of length up to 30. There's something in between. Yeah, so this is length 30 to 150, and this is what I call in the meantime, what many people call in the meantime, is the twilight zone. Yeah, and calling deletions and insertions still, I mean, Peter mentioned this also. Still, it's a difficulty to call certain types of structural variants, and this is one class of them. Yeah? So my contribution to this was to develop a method that back at that time, 2014, I was a member of the consortium of the Genome of the Netherlands project, and here comparing with the phase 2 variant count plots that the 1000 Genomes project had gathered. There was a ditch here which is right in the twilight zone. They hadn't discovered any. And we are the first ones to discover, like many, at a validation rate of 98%, of course. So these are all high quality variants. They were the first ones to fill up the databases. So I am an expert for this kind of job. Yeah? It was my contribution to the project based on this method here. So let's have another look. So this is all germline variation. So the history lesson part 1 is over. Germline variation is difficult for certain classes of variants. because due to the uncertainties affecting the data. Yeah, and well, then the second part is okay. Now we thought, okay, if you look into somatic variant databases, yeah, then what you see is still, you still see this, you know, ditch here. Yeah, I mean, there's no variance whatsoever discovered in that size range in terms of somatic deletions and insertions still today. Yeah, maybe Jan Koppel knows a little more, but I think hardly any. Huh? I'm pretty sure. So we thought, why not remove the twilight zone ditch also for somatic indels? Yeah, so let's do that. However, we ran the tools and we didn't succeed. Yeah, of course we did not, because you have to deal with another level of uncertainty for somatic variants. Yeah, and the problem is that is the heterogeneity of cancer, the clonality, and so on and so forth, and the low allele frequencies you are dealing with, and so on. Huh? And also just in general that you, okay, this is the story. Yeah? So here you have a healthy bulk, and then the allele frequency, in my case, this is the VAF from Peter, the variant allele fraction. Yeah? So this is the probability that given a certain variant locus, you sample a fragment that is affected by variant. Yeah? So, and there's three possible choices, there are three possible cases in healthy genomes is the probability is either 0, meaning the variant does not affect any of the, has not been transmitted neither by the father nor by the mother, so it doesn't affect any of the fragments in your sample. Yeah? It's 0.5 if either the mother but not both, or either the mother or the father but not both transmitted the variant. So the probability to sample a fragment is 0.5. That is affected by the variant is 0.5. It's not exactly 0.5, of course, as Peter pointed out, because you have some fluctuations, but still the truth is 0.5. Yeah? So, or it could be 1, meaning meaning that both ancestors transmitted the variant to you. And then with probability 1, you sample a fragment that is associated with the variant. In cancer, we're dealing with these kind of pictures, and all of those, I mean, the colors indicate different clones, and all those different clones are affected by different kinds of somatic variants. And also you have some healthy cells among them, just the blue ones here, and more than that, we do not know the picture, of course. So imagine for a while we know the picture, just to help you out of this. We can imagine, for example, that we are dealing with a heterozygous variant affecting all heterozygous somatic variant affecting all the tumor cells. Yeah? Then you have like an allele frequency, a VAF, of 0.5 times 0.75, because 3 out of 4 are cancerous, and this makes 0.375. And you can go down, of course, well, really low. Yeah, in this example here, you end up with an allele frequency of 0.09375. So summing it all up, you have in healthy samples, the truth is it's either homozygous, heterozygous, or not present. In cancer, the truth could be anything. Yeah? In a way. Some things are rather unusual. So somatic variants usually don't have a variant allele frequency of like approaching something 1, but I mean still, who knows? Huh? So there's another level of uncertainty here, yeah, induced by those pictures. So let's have another look at the data we're dealing with, and imagine that someone told you, okay, so there's maybe a deletion here, and then re-examine the data that gives that may provide evidence of this deletion to occur. Consider this is a picture. Actually, this is certain data. Everything you see here is true, yeah? Meaning all the alignments are correctly placed, yeah, and you also know that the red ones are actually associated with the variants, yeah, and the gray ones are not. So figure this is from the control genome, and then of course you're dealing with a heterozygous variant because one of the ancestors pass on the gray fragments to you and the other one the red ones in a way. Huh? Whereas in cancer you already do not really know, so all you can derive here from this picture in a straightforward account is kind of you can compute the maximum likelihood estimate of the AF, which is 4/7 here, because 4 out of 7 reads were affected by the variant. Yeah, that's all basically you can do, and you have a certain probability distribution of over the different allele frequencies, which is governed by a binomial distribution. Huh? So here, I mean, in control you have full certainty. Given full certainty of the data, you have full certainty about what was the case. In cancer, you already with certain data, you only have a probability distribution over the different kind of allele frequencies that could be true. Okay, and then we have the old uncertainties. Have the reads been mapped to the right place? And if yes, do they really support the deletion reference allele? Meaning, if yes, are they red or black, gray? And so everything you can do is kind of compute probabilities for certain scenarios to be the correct ones. So for example, this you can compute a probability for this scenario being the right one. So all the reads you see are correctly mapped, and those 4 are red, meaning they are associated with a variant, and the other 3 are not. What you can get from the aligners is you can get probabilities that tell you whether the reads really align correctly, and you can also make use of probabilities that also delivered by variant callers and aligners and so on and so forth that tell you how likely it is that a read stemmed from the deletion allele. Yeah, so you can compute those, yeah, independently of what I'm telling you here, and you can make use of this, and so you can compute a probability for this scenario to be the correct one. Yeah? This computation.
Sorry, I just want to check if I'm following. So in this red stuff, this can correspond to like a population of cancer cells where this has actually happened, and then you are explaining this that you don't see this in the non-red stuff because it could be like normal cells where the deletion didn't.
Yeah, it could be all kinds of things. I mean, in cancer it could be this is 1 or 2 clones affected by the variant, or you don't know how many, right? And then this here could be healthy cells or other clones that were never affected by the variant. So all kinds of things, yeah, all kinds of scenarios are possible. You do not know. Yeah? Okay, so also this scenario could be the correct one. So reads 2 and 4 and 6 were not correctly placed. 1, 3, 5, 7 were, and then you can compute a probability for this scenario to apply as well, and so on. I mean, this here now the uppermost is also, it's unlikely to reflect the deletion, but still, yeah. So it has probably non-zero to be red. Huh? Anyway, so now think about this for a while. So how many scenarios are there? So either every read could be either not there, gray, or red, reflecting it's not correctly placed, or correctly placed and reflects the variant, or correctly placed and does not reflect the variant. This is 3 cases. So you have 3 to the number of alignments you're dealing with, and then if you are let's say coverage 30, which is a minimum in a way, usually you sequence cancer genome as 60 to 80 and control 30 to 40. So 3 to the 30, 2 to the 30 is a billion, 3 to the 30 is even more. So you have you deal with a billion more scenarios, and this is a mess. Yeah? So a statistical mess. You need to be you need to do something about this. Yeah? Why? Because you would like to compute the following thing. Yeah? You would like to compute a likelihood function that a certain allele frequencies apply given the data you observed. Yeah? The alignment data from the healthy and the cancer sample. Yeah? Theta sub h is the allele frequency in the healthy sample, in the control sample, and theta sub c is the one in the cancer sample. And then you get data from alignment data for the healthy sample and the cancer sample. and then you would like compute this likelihood function of the two variant allele frequencies to apply given the data observed in the two samples, in the two genomes. So if you are in control of this function here, you can integrate over this, and then you can compute a probability that given the data you see, you observe the variant is indeed somatic. So here, the likelihood here is the healthy allele frequency is fixed to 0, reflecting that the allele frequency in the healthy sample should be 0, meaning, in other terms, that the variant did not show. So it's supposed to only show in the cancer genome, otherwise it's not a somatic variant. So this is why I fix it to 0 here and then integrate it. And for being able to integrate this, I need to evaluate it this here at several points, so I need to be really quick. I need to be able to compute this quick. Okay. Yeah, that's what I told. And now consider, okay, we have like, we just do this in a naive manner, then we have to sum over all possible scenarios. So this is the computation, fairly straightforward. However, you have to sum up over all scenarios. Every phi indicates a single scenario, and this is a billion scenarios at the very least. So you're not able to do that. At least you're not able to do that like a million times along the whole genome for every variant locus you encounter. So you need to be clever a little. Yeah, this is what I told. One more time. You need to compute this efficiently to be able to do this integration and also, and then be able to really soundly quantify the probability that a certain variant is somatic. Okay, so this is what we came up with. We basically just sat down, Math Institute, of course, so just sat down, pen and paper, and then we just figured out how the different variables we're dealing with here are connected with one another. So meaning in statistical terms, we figured out what are actually, what's the reasonable set of hidden variables we're dealing with, like variables which we do not observe, and what's the observed data we're dealing with, and then how do they interact in terms of statistical dependencies. So first of all, in circles, you have the hidden variables you do not know, you cannot immediately observe from the data, and the rectangles are the observed data. So first of all, the most important one of those hidden variables is the allele frequencies. This is the one you would like to estimate. And then omega I is a binary variable that reflects whether the read I was correctly mapped or not. And here, every column reflects one alignment. So you have about like at least 30 to 40 here such columns connecting to the allele frequency. And every column has the observed data, which is either X when you have a paired alignment where the possible variant affects the internal part. So the alignment length is an integer. Or you have if the sequence part covers the variant locus, then you have you're dealing with a binary variable which indicates whether a split is present or not. And you have the omega i's correctly mapped or not, and you have the xi i's which reflect whether the read indeed stemmed from the deletion allele or not. So one for it's affected by one means red in the earlier pictures. This being 1 is red, and this being 0 is gray, and this being 0 means it's not there, and this being 1 means it's there. So this is fairly simple, right? You just put this on a piece of paper, and then you go on thinking about this. And okay, so you also have to specify all the probability distributions that govern the hidden variables and the observed data and also the dependencies. So for example, as an example here, so this arrow here, these two arrows, this is here the alignment length. Given that the alignment is correct, and given that the variant is not associated by the, the alignment is not associated with the variant, then this is governed by the fragment length distribution. Yeah? If it is correct and if it is affected by the variant, then it is governed by the fragment length distribution shifted to the right by the length of the deletion, which is delta here in this.
Is that normal density? Just like Gaussian density?
It's near normal in many. It doesn't really matter. In my approach, it doesn't matter. You can deal with all kinds of stuff. I mean, it matters in some approaches. In this, it doesn't. Yes. I mean, okay, in many cases it is anyway.
So you don't assume any specific distribution? You don't assume any specific distribution?
No, not in this approach. I did this earlier in Germline variant calling, but I mean here it's not necessary to assume this. It's just really generic. Okay, so then if you have another look at the model, you see, okay, you can compute the likelihood function actually in time linear in the number of alignments, which is just the best you can get. It's just by the proof is done by exploiting conditional independencies pointed out by the model I just showed you. It's pretty straightforward, and you can also prove that the likelihood function actually achieves a global optimum somewhere. Which is nice. This is obviously the maximum likelihood estimate that you are aiming at. Yeah. Okay. So as for results, so we had some nice simulated data. The genomes are real in a way, but we simulated the reads, and then what we do, we have a tool which is called ProSig, processing somatic indel calls. Or post-processing. Yes.
So this
Sorry.
model assumes all the reads are independent, right?
Indeed, yeah.
So then okay, then it is linear. And aren't they actually, like, because some of them overlap, no? Like in the alignment, they overlap.
Now given, the model assumes that given the allele frequency, they're independent, and this is a reasonable assumption, yeah? So if you assume you know the allele frequency, then that's a realistic assumption. Yeah. I mean, considering the variables I'm considering here.
The allele frequency, then some reads map in a similar place, and doesn't it make them?
Yeah, but given you fix the allele frequency, then you sample them independently of one another. Yeah. So you sample one, you break out another fragment, and so on and so forth. Yeah. So it's kind of well, breaking out a fragment here. Well, maybe the minor effects, I think. So in that respect, you're right, but I mean, otherwise it's fairly independent. Yeah. Okay. So here, so the tool we now have is kind of, okay, some other tool gives us calls, and then we re-examine the calls and go back to the data and re-examine it. So we did this with state-of-the-art callers. We used Deli, Lancet, Manta, and Strelka. We also did this for Platypus and Pindel, but Platypus doesn't really look good for Platypus. For us, well, but Platypus is extremely conservative, and Pindel is terrible. Yeah? I'm friends with the author, so, but I mean, it's just not, doesn't work. You just cannot use Pindel for semantic variant calling. We can.
I didn't understand what you're doing now. You compute the maximum likelihood estimate of theta.
Well, this is one part of the game. The other thing is, okay, I can go back.
What is ProSig now? What's input and what's output?
Yes. I mean, we compute this probability for a variant to be somatic given the data. Yeah, and then we
like
yeah
so
yeah,
okay.
and then yeah, I forgot to tell. And then what we can do is we can actually, and I think this is kind of a novelty actually. We can control the false discovery rate. I'll give you some more plots with this. But I mean, then the callers give you points. So Deli and Manta and Lancet and so on, they give you points, which are usually very conservative. So they have high precision. So this is the recall on the x-axis. This precision. So recall is the fraction of variants discovered you should have discovered. Yeah? And the precision is kind of the fraction of calls that are correct. Yeah? You made. Yeah? And so usually the callers tend to be very conservative, so they operate at high precision, but the recall is rather low. And if you apply our tool to that and you re-examine this and you re-evaluate this and also play the false discovery rate control game we can play now, you can drive the recall much further out. Yeah, so from here, this is the twilight zone finally. Still, we can also do some good here, even in the areas where, I mean, I told you there is another level of uncertainty here affecting somatic variants. So also easy calls are affected by this. And also Peter showed like, okay, unfortunately, I do not have any plots that reflect what the distribution of the allele frequencies were in our simulated data, but they can be very low, and I think we can improve a little on your numbers, but I'm not sure, of course, because I didn't really compare with them. But still, well, the uncertainty really grows when you, when the allele frequencies drop. Huh? I mean, but what we can do in this case is still we can give you probabilities of this thing to occur. You might not be inclined to call it, but still we can tell you, okay, there is a variant of allele frequency, say 0.5%, and the probability that it's really occurred is kind of 40%. Yeah? So still this, yeah? While nobody might be inclined to call it, we can quantify. Yeah? So which can help you in other ways. I tell you later. Okay, so here, this is the story. We can enhance sensitivity in discovery significantly if you run our tool on the call sets from the others and re-examine data. Huh? Also, false discovery rate control, which I claimed was a novelty in a way, is near highly accurate. Huh? It's part of the game. Anyway, the allele frequency estimates are also good enough, say, yeah, reliable. Let's go on. This is the tool. I mean, the first author in the meantime is Johannes, formerly Dana-Farber, and then a postdoc in my lab. Okay, let's do the last part. Yeah? So how to call SNVs in single cells. Yeah? I'm not a. Don't I have like? Give me 7, 8, okay?
Okay, 6.
Yeah, but okay, you're dealing with amplification biases on top of all the things I told you. In SNVs, actually, read placement and variant certainty is not that much expressed, but still, yeah, it might matter in single nucleotide variant calling. It might matter. Yeah? So here I considered multiple displacement amplification in all of the data I was dealing with. So which Peter pointed out, is this the method to choose or is the method of choice in a way? But still, let's go there one more time. So here you introduce amplification biases. You may have you encounter a lead dropout. So you haven't sampled any of the material in certain places. In others, after amplification, is heavily distorted. Yeah? So due to that, you have false negative alleles reflecting dropout, yeah? And distorted allele frequencies reflecting kind of amplified parts. Huh? And then also you might introduce polymerase copy errors, or heat lysis errors, or so you may also encounter false positive alleles, yeah? After amplification. Huh? Okay, so we have to deal with different terms here, and I would like to make use of that and try to make use of my models from before to deal with single cell data now. And I need to clearly distinguish between the allele frequencies before amplification, which are either, even for somatic variants, are either 0, 1/2, or 1, yeah, because you only have 2 copies. Yeah? So this looks kind of nice. However, after amplification, everything is possible, yeah? And it's completely distorted. How to deal with that in terms of the models I just presented? Okay, let's run through this picture here. So we have a bulk, and we have like somatic variant like C instead of A affecting some of the cells. Yeah, in fact, if affecting 1/8 cells. Yeah, if you sample some bulk, then you see, okay, you encounter this in 1/8 cases. And here you sample two single cells, one of which is not affected and the other one which is affected. Yeah? Okay, then you amplify and then you distort it. You introduce a polymerase copy error, whatever kind of error here, and you go from theta being 0 to rho being 0.2. Rho is the VAF after amplification. Yeah? And this is 0.5, but after amplification, this is only 0.14, although, I mean, this is lower than this one, yeah? So this is a problem. And this is somatic variant, and this is not. Yeah? So how to distinguish this? We consider this paper, I think Peter was one of the authors here, and they present some nice probability distributions that given a certain theta, like 0, 1, 0.5, what is the distribution of the rows afterwards, yeah? After amplification? This is easy, 0 and 1 are easy, but 0.5 is difficult. Yeah, so you see this is kind of near uniform afterwards. It's terrible. Huh? So we can apply, we can make use of these distributions here to complete our statistical account, and then we compute some likelihoods for the different cases. So it's 0.6 in this case, which is higher than here, and here you might conclude, okay, there's no variant actually, yeah? Whereas here you might conclude, okay, there is a variant, although this is not correct. But what I definitely recommend is to make use of the bulk, because this is a lovely control element. Yeah? So because even, and this is the story, even if you're not inclined to call it because it's so low frequency, yeah, still our model gives you a probability for this to occur, and then you can combine this with the probabilities inferred here, and then in the end, yeah, given some minor, okay, if you have a variant that for which you're inclined to call it at a certainty level of say 20%, so nobody would call that, right? It's like it occurs in the bulk at an allele frequency of 2.5% or something. Still, there's some evidence, and you're inclined to call it at 20%, say. Yeah. Nevertheless, if you then mix this with the evidence you raise from the single cells, you might be helped. Huh? And then in the end, you see for certain, I mean here is 12.5%, but if you then in the end you end up with the right conclusions. You call this here, but not that one. Huh? Okay, so this is the model. Actually, so the whole model from before applies if you replace theta with rho in single cells, and then I can accurately estimate whatever I want to do, determine these rows and determine the probability distribution of the rows given the observed data. Yeah? And then I need to make the step back to the theta using the beta binomial distributions from published in Lodado et al. In 2015 in Science. Huh? And then in the end, what I have is kind of I can give you a statistical account of, I can compute probabilities. I run my bulk model, I run my single cell model, and then with kind of juxtaposing lots of cases and so on and so forth, I can give you probabilities for like dropout to occur, amplification errors to occur, events to be somatic after all, and this is what we're all after, right? And then and the point is just really to really have a nice statistical account of the uncertainties that affect the thing. Results, we have real data here, so this is really, really lovely. We invested a lot of time in making this data set. I cannot tell you how it is worked, but still, this is a nice data set. Really, it's real data. Yeah. And here we compare with, we can still make use of the false discovery rate control, which other tools cannot do. And then we compare with the ASCII and the Monovar and ProSolo, which is our new tool, is kind of, you know, yeah, again, increasing the recall. And if you zoom in this upper left corner, then you see also we also at the recall rates that those achieve, we also outperform them there. So obviously the whole thing when it looks really stressful, and you have to do all the statistics and all kinds of things. But in the end, you're rewarded. So I encourage you to to do some mathematics and computer science after all. Huh? Thank you very much. Huh? Okay.
A wonderful take-home message. Other questions for Alex? But still, we have time for 2 burning questions. Well, now you put everybody.
No, you can ask the question.
I don't mind. I really, I don't care.
Please ask the question.
So, can I ask about, in the first part, what was the trick? Was the main trick to use these uncertainties of the alignment that the mappers spit out? Is that in the end where new information comes in mainly? Or what would you say? What's the main trick?
The main trick for the somatic index, yeah, you mean?
Yeah. I mean, I understand there is a model and so on, and everything is important.
Yes,
but
yes.
Intuitively, what's the main thing?
Intuitively is indeed that yeah, when I mean, this is really data heavy science, yeah, in a way. So we really do our best to use what you get from the alignments in terms of parameters and whatever Fred scores and all these kind of things, which are deemed to be reliable, and in fact they are if you use them. In many cases, but we also do some realignments. So part of the game is also to realign here and there if you're not entirely really sure, and then also introduce the variant into the genome and then remap. So this is also part of the game in the meantime, and then we improve on the statistical counts given by the aligners. So in this way.
Thanks, Alex.